So my lecture will be talking about these two aspects using two different genes and with the associated syndromes. So let's begin. As we all know that clinical and research genetic studies has accelerated the pace of identification of disease genes. And this is due to the advent of NGS-based technologies, which is primarily focusing on whole exome sequencing and whole genome sequencing. And the field that has benefited the most is the identification of de novo variants with this NGS-based technologies. But what are the confounding factors of increased variant detection by WES? So for example, you have identified a de novo variant through WES. And just looking at the segregation in the family, that though both the parents do not carry the variant and the proband is showing the heterozygous in the Sanger, is not sufficient to call it as a pathogenic variant. You need more families, you need more missense variant in order to call that particular gene as the de novo variants implicated with the disease associated. So novel disease genes represented by the few cases and the evolving characteristics of the variant detected, which is increased incidence of the de novo variant. And then what is the challenge? The challenge comes in translating these findings into clinically actionable findings and basic science clues. So these are the characteristics of clinically actionable pathogenic variants. So for example, null variant in a gene where loss of function is a known mechanism of disease. Number two characteristic, same amino acid change as a previously established pathogenic variant regardless of nucleotide chain. The prevalence of the variant in affected individual is significantly increased compared with the prevalence in the control. And the well-established in vitro or in vivo functional studies supportive of a damaging effect on the gene or gene product. So if we categorize them, so the bold letters you can bold, you are seeing that it is categorized into two forms: functional analysis and genetic evidence. So if you have identified a novel pathogenic variant in the clinic, so you have to undergo two categories, that is genetic evidence and functional validation, which are required for making clinically actionable discoveries. So let's go on to the genetic evidence first. So as per the ACMG criteria, if you want to have more genetic evidence of the variant identified in the clinic, you have to go for number of additional variants that can be identified in the gene, and how you can identify the additional probands through online different web-based portals, that is Gene Matcher and Matchmate Exchange, Decipher. You can use this. So I am sure that you are all aware with the Gene Matcher. So where you are putting your patient's information, and then you are brought into you are bring into the communication with other physicians and researchers, where you can exchange the phenotypes, and it depends on which stage of your study is, whether it is in the human genetics stage or it is in the functional stage, and then how they want to collaborate and so on. Second is consistent genotype and phenotype correlation, which will talk about variant characteristics. So the variant characteristics means like whether it is a missense, truncating, and zygosity versus the clinical features. Then under this, amino acid and protein domain versus the clinical spectrum of the features. Under the functional validation, we want to test what is the mechanism of pathogenicity, whether it is undergoing the loss of function, nonsense mediated decay, or gain of function. So if we want to study the functional validation, what we would be looking for, impact of coding variant on the gene. So here, variant type, whether it is a truncating, splice, or missense variant, whether the protein is undergoing nonsense mediated decay or it is stably expressed, and where is the what is the location of the variant in the gene. These are the three segments in which you can identify the impact of the coding variant on the gene. Now you want to identify the impact of coding variant on protein function. So how you are going to do it? Whether you will be looking for disrupted function. So disrupted function means that if there is a mutation, then you are seeing the changes in the function of that protein. Whether it is necessary or insufficient, whatever you are doing the experimentation, it is actually necessary and sufficient, or it is going to correct the variant or rescue the phenotype. Whatever experimental plan you are doing for the variant of interest, does it actually make sense? So you want to proceed through sanity check as well. So in the first case, I'll take the example of CYT and ASXL3. So to start with CYT, that is citron kinase. So here we have the cohort of developmental disorder in which microcephaly was one of the clinical feature. And by performing using the combination of whole exome and the genetic evaluation tool, we identified a splice variant in the family A. So now we want to perform the genetic and functional validation since we started with one family in hand. So we went on and pasted the information on the gene matcher to identify additional variants in the same gene. And to the surprise, we identified truncating citron kinase variants in two different families, as highlighted in this figure with the Sanger traces. And here we saw that the truncating citron kinase variants were having more severe microcephaly when they were compared with missense variants in the citron kinase. So these are the schematics of the pedigree and the Sanger traces for the citron kinase family affected probands. So our first genetic evidence is checked. So we started with one family in our hand, and through gene matcher, we identified 6 families. So from the human genetics, we are very sure that citron kinase is the genetic basis of the primary microcephaly. So one is checked out. Now we move on to our second genetic evidence, that is consistent genotype and phenotype correlation. So here we want to see what type of variants we are identifying in the gene. Is there a consensus of the phenotypes and the variants in the gene, or they are different? So we kind of mapped all the identified variants in the citron kinase domain architecture, and we found that all the variants were localized to the specific protein kinase domain, as you can see over here. And there was one splice donor variant that was in exon 9, and that was also very close to the protein kinase domain. So here it is very much clear picture that we are seeing the localization of variants in the protein kinase domain, irrespective of being missense, truncating, or the splice variant. And we have one more family, that is family C, which had a compound heterozygous variants, in which one was the truncating, another was the missense. And in this family, the clinical features were kind of intermediate. So we are seeing a graph of truncating giving more severe phenotype, missense giving moderate, and the compound head giving the intermediate phenotype. If the compound heterozygous does contain the truncating variant. So our second evidence and second genetic evidence category, the first part, that is also cleared now, because we can see that the variants are localized and how they are giving the schema of the phenotypes. Now we want to go for the amino acid and protein domain versus the clinical spectrum of phenotype features. So we see that they are all localized to the protein kinase domain, and the clinical spectrum, as I explained in the previous slide, that there was the schema of the microcephaly clinical features. So we move on. So genetic evidence till now it is completed. Now we move on to the functional validation. That is the mechanism of pathogenicity. So here we want to look at impact of coding variant on gene. That is the variant type, whether it is truncating, missense, splice. and whether it is giving the nonsense mediated decay or it is giving you a stably expressed protein. So to look at this, in this figure, we have all the three. We have splice as well, we have missense, and we have truncating variants. So to this particular segment, we will say, yes, we are seeing all the variant type. Now we explain this, whether we are seeing stably expressed or nonsense mediated decay protein. So to this, we performed a in vitro assay, that is mini-gene assay. So for this, since patient cells were not available to us, so we cloned the segment where the mutation was present in a vector and transfected that vector into the cells. And then so the vector was already containing a reporter exon. So imagine a situation that when we will transfect the vector along with the region of our interest, which has the mutation, so the cellular machinery will come and do the splicing. So if there is a mutation, because this is the splice variant we are characterizing, so we want to figure out if there is a mutation, whether due to mutation we are seeing either skipping of exon or there is a portion of retention of intron. Because once we tell this, yes, because of this splice variant, we are seeing skipping of exon or retention of intron, then we will be able to say, yes, this splice variant is pathogenic. Although there are computational tools, but still we need to tell them by performing these types of mini-gene assays. So with this experiment, so over here, these are the inbuilt exon in the vector. So this is an empty vector. And this is the exon 9, the wild type, which has no mutation. And this is the exon 9. And the green asterisk shows the mutation of our interest. This one, mutation, which is G2A. And when these vectors were transfected into the cell line, the cell's splicing machinery came and they performed a splicing event. And then we extracted the RNA, make the cDNA, and did the RT-PCR. And you can see over, you can appreciate over here that here we are seeing the retention of the intron as a major product. And we can definitely see the protein, the mRNA transcript, the cDNA here is stably expressed. So with this, and this is the here, exon 9, after this, the Sanger traces are showing there is a retention of the intronic region. So with this, we will say that the protein is stably expressed because we were seeing the traces of those transcripts. Now we want to look at the location in gene. So we know that all are localized to the kinase domain. So with this small experimentation, you have identified two things. Number one is that you identified, yes, this splice variant is pathogenic. And number two, why it is called pathogenic? Because with this splice variant, there is a retention of intron, which is leading to stably expressed protein, which is actually leading to the citron kinase machinery affected disruption, because of which we are seeing the clinical features on a very basic level. Then we wanted to look at what is actually the published gene function, because we did our experimentation, but we wanted to look what is actually known about it, because we started with a novel gene. So we looked at and we found that citron kinase localizes to the cleavage furrow and is required for cytokinesis completion. So if there is a mutation in citron kinase, definitely you will be having a binucleated cells as opposed to a true mother and daughter cells, as similarly shown by immunohistochemistry assays. So here, since all of these variants are now localized to a specific domain, and if you have all the in vitro assay system, what would be the next step you would be choosing for this? Like you have you know, these are the variants that are localized in the kinase domain, and you have all the well-established infrastructure for the lab. So what you would be interested in knowing?
What is the function of this variant effect?
It is in the kinase domain. So yes, we know it is in the kinase domain. So what definitely you would look for? Function. You want to say function.
For the next, other substance would be affected by this kinase.
Other substance would be affected by, would you want to look for the kinase activity?
Yeah.
Because you are seeing the mutation in the kinase domain. So would you not be interested in looking for, if they are in localized to a kinase domain, then what would be the kinase activity of this particular region where all the mutations are lying, isn't it? That's how you would be interested into, or anything else you would be interested, isn't it? So would you be willing to clone the whole fragment of the gene or just the part of it? Just the part of it, isn't it? When you will be looking for the whole, when you will be doing for the whole segment of the gene, when you would be interested in looking for the whole segment of the gene? When the mutations are scattered, then you will be looking, and what else you could be doing? You want to look at the more like the protein folding strategy. If there is a mutation, then how it is folding and how it is affecting its the neighboring components as well. If your question would be that, then you would be cloning the whole fragment and overexpressing. You want to identify the binding partners of citron kinase because it's a novel gene in your hand. Nothing is known about it. There is no literature. You will just find one, say, zebrafish model or some lower organism. So you would be very much interested in how it is interacting, what are the partners are there for this gene. Or since it's a citron kinase, so it's an enzyme based. So what is its substrate? Because till now also there is no known substrate for citron kinase. So here we will be looking for kinase activity. So for this, we cloned a fragment of this citron kinase gene along with the mutation of our interest that were created to site-directed mutagenesis because we did not have the accessibility to the patient cells. And then we looked for the, using the Gomasi staining, we looked for the kinase activity, and we found that the kinase activity was actually affected in the case of the missense variants. So now we come on to fill the blanks that we have started with. So the truncating variants was affecting the structure and kinase activity, and the missense variants were mainly affecting the kinase activity. So whether we are performing all the experiments or if we can correct the variants, yes or no. So we want to look at that as well. So we went back. We created we used homology directed repair as a mechanism to correct the variant, and we looked at the cell lines, like how they were and here we want to characterize whether we are seeing the binucleated nuclei in the case of citron kinase mutant type, and when we are correcting the variant, are we also still seeing the binucleated nuclei? So we kind of saw it was the phenotype of the cellular binucleated nuclei was rescued when we performed the HDR based genetic genome editing. So I would say, yes, it is necessary and sufficient to either introduce or correct the variant that have been identified in citron kinase. Does everything we are doing, does it actually make sense? Is it actually going to contribute to the field of the science? So let's check that out. So as we all know that primary microcephaly has a genetic basis, and till now many genes have been identified as a genetic basis of primary microcephaly that can be identified during either at the birth or before that. And over here, citron kinase position, which is present in the cleavage furrow constriction, it makes a very interesting finding that yes, it is actually contributing to the phenotype of primary microcephaly. So to this, I will say yes. So does anyone have any questions for this part? Is it very clear? like how you will go from genetic evidence to functional validation step by step, and what actually you would be looking for when you are performing the either genetic evidence based categorization of the variant or the functional validation. So this was the case of a splice variant. Now I'm going to take the example of de novo heterozygous variant, and that is the truncating variant. So when I started the lecture, I told that what are the like which variant actually got benefited the most was the de novo variants with the whole exome sequencing. And this is kind of a nucleosomopathies where you can identify the variants in the genes that encode the components of proteins that can read, erase, and write. That means post-translational histone modifiers. And to this, there was, in the clinic, we saw a patient with a 10-year female seen in genetics clinic since birth. She had the complaints of intrauterine growth retardation, failure to thrive, severe global developmental delay, hypotonia. She was unable to sit, crawl, or walk unassisted, delayed gastric emptying, and GT feed since 8 months, and there was a craniofacial dysmorphism. She was still non-verbal at the age of 15 years. She had seizures, severely disrupted sleep patterns, and left-sided estropia, hyperopia, and astigmatism. And she has 10 degrees of right-sided scoliosis. This was the brain MRI at the age of 1 year, where you can see that this is the thin corpus callosum, and there was a cortical atrophy. So this case was actually studied when back in 2013 and 14, when there was we have to perform the series of genetic evaluation before we order for whole exome sequencing. So this proband underwent methylation testing for Russell-Silver syndrome, methylation testing for Angelman syndrome, sequencing and deletion duplication analysis for Robin-Roy syndrome, uniparental disomy testing for 11p15 based on her clinical features. And then finally, when everything came out to be negative, she was sent for whole exome sequencing. So in the whole exome sequencing, this de novo variant in ASXL3 gene was identified, and this was identified as a VUS. So we started with as VUS, and this was the frame shift variant, which was threonine at 484 position. That leads to frame shift after 5 amino acids, and the parents tested were negative for the variant. So we went for the genetic evidence. We wanted to identify additional probands through different portals. And by that time, when we started out, so we identified in 2013 September. In December, this paper came out for the BRS, that is Bainbridge-Roper syndrome, where now it has been more populated with the variants in ASXL3 gene. And now we know that ASXL3 as the genetic basis of Bainbridge-Roper syndrome. So we went for consistent genotype-phenotype correlation. And this is the color coding over here, shows the penetrance scale, where the red shows the 100% penetrance across all the affected individuals. That means that global developmental delay, intellectual disability, autism, failure to thrive, hypotonia are the key clinical features of the Bainbridge-Roper syndrome, followed by the seizures, which has a reduced penetrance. So we wanted to know what is the published ASXL3 function. So we drew a schematic where we found that ASXL3 has the domain ASXN, ASXH, ASXM1, M2, and a PhD domain. And it belongs to the family of ASXL that has the partners so as ASXL1, ASXL2. And ASXN is a predicted forkhead box DNA binding domain. ASXH, M1, M2 are epigenetic scaffolding protein-protein interaction domain. And the PhD is the histone post-translational modification reader domain. And which is interesting is that ASXL germline variants have been linked with various syndrome. That is, ASXL1 causes Boring-Opitz syndrome, ASXL2 causes Shashi-Pena syndrome, and ASXL3 causes as the genetic basis of Brain-Bridge-Roper syndrome and autism spectrum disorder, whereas somatic variants have been linked with various cancers. And in the year 2010, it was identified in the Drosophila that the ASXL forms a complex with the BAP1, which is a polycomb repressive deubiquitination complex. And this complex is important for post-translational histone modification. So this is the nucleosome, and this is the histone tail, where there is a ubiquitin mark here. When I'm talking about ubiquitin mark, it is more with regard to post-translational histone modification, and it is a repressive mark and not the mark of protein degradation. So it is more towards epigenetic model. So this component comes and decorates the ubiquitin mark on the tail, whereas this component, which has been identified, ASXL1 and BAP1, this comes and this removes the ubiquitin mark. And this ASXL1, all of the three members can interchangeably act in concert to remove this ubiquitin mark. And this dynamic balance of decoration of ubiquitin mark and removal of ubiquitin mark is important for transcriptional regulation. So first of all, I wanted to see what are the binding partners for ASXL3, because it's a novel gene. So I cloned the full length ASXL3 gene in a flag vector, and then did the co-IP, like immunoprecipitated that gene with the antibody of interest, and I found that ASXL3 was actually interacting with BAP1. And ASXL3 BAP1 interaction is important to mediate the removal of ubiquitin mark, although ASXL3 is not an enzyme, but its concert with the enzyme, because BAP1 is an enzyme, its concert with the enzyme is important to remove the ubiquitin. So now we want to look at the variant type, truncating, missense, and splice. So here you will appreciate all the ASXL3 variants reported till date are all truncating variants, whereas missense variants and in ASXL3 has been implicated in autism spectrum disorders. So we went to look for the genetic basis, like what is the genetic mechanism of ASXL3. So we had the patient skin biopsy differentiated to the primary fibroblast, and this patient was threonine to asparagine, which leads to frame shift after 5-amino acid. And so we extracted the RNA, looked for the cDNA, and then we look for what is the transcript. And over here, and at the same time, we cloned the full length of the ASXL3 fragment and transfected into the 293T cells as well to see its effect, whether it is expressing or not. So you can see over here that in the frame shift, there is reduced expression of the ASXL3. Similarly, when we quantified the mRNA level, you can appreciate over here that there is a significant decrease in the mRNA transcript of ASXL3 in the fibroblast cells. And similarly, these are the results for the 293Ts and primary fibroblast cells. So with this, we were able to say that the ASXL3 variation leads to nonsense mediated decay. So, but have anyone seen ASXL3 variants towards the C-terminal? Are they being identified in the C-terminal? Like so these are the variants. If you see over here, exonic structure, they are more towards in exon 11 and starting of exon 12. This is the domain architecture. So what would be the, would you be willing to say NMD when it is more towards the end of the C-terminal and you are still watching, you are still seeing the clinical feature of Brain-Bridge-Roper syndrome? You would say. But there are specific points to consider that you can tell whether NMD can happen or not. So towards the last exon, 50 nucleotide from the stop, it should not lie in that in order to, because then it will not, NMD will not happen. And usually if that is the last exon, and if you have the variant in the last exon, either it should be at a very starting of the last exon, so that then it can process with the NMD. But this last exon is very confusing when there are very strict points to consider when to actually say yes, NMD will happen or no, NMD will not happen. So in that case, we do not know whether NMD will be a genetic mechanism for the same yes or no. So we actually have to repeat all the experimentation for that exon. So interestingly, we have the variant which is in ASXL2, whose genetic mechanism is also nonsense mediated decay. And recently we have identified one variant which is in the last exon in the C terminal, very close. So I have to test it in the lab now. What does it say? So here we want to look at the location in the gene. So it is not very localized to a specific region as we saw in the case of citron kinase. So it was all like across the gene. So now we went on to the impact of coding variant on the protein function. So over here, we know that this is the polycomb repressive complex that comes and decorates the ubiquitin mark. And then this is ASXL3 with the BAP1 complex that removes the ubiquitin mark. And this dynamic balance is important for the transcription regulation that allows for Hox gene patterning. That means to have the head to tail development. And similarly, we have shown with the ASXL3, we have the truncating frame shift variant, and we show the genome-wide ubiquitination level in the case of patient, it was increased. And similarly, as tested to the S3 mRNA transcript level. We went back and ran the RNA sequencing and looked for the differentially expressed gene in the patient fibroblast, and we found that there were differentially expressed genes in the bulk RNA sequencing. So the disrupted function that has linked ASXL3 mechanism for Bain-Bridge-Roper syndrome is H2A deubiquitination gene expression. So next we wanted to look for H2A monoubiquitination dysregulation as a key Bain-Bridge-Roper's syndrome molecular pathology. So we went back and created patient-specific cells through CRISPR-Cas system through human embryonic stem cells. And then we neural differentiated those to NPCs, and we still identified the similar results. That means H2 monoubiquitination levels were highly increased in case of heterozygous state. So we can say now confidently that it is the elevated H2 monoubiquitination level which is leading to the phenotypes, clinical features of Bain-Bridge-Roper syndrome. So was it necessary and sufficient? Yes, both, because we kind of introduced the variant and looked for the molecular pathology. So sanity check, does this all experiment actually make sense? So yes, then I kind of summarize all the findings, and we saw that H2 monoubiquitination regulatory axis is important and has been implicated in the neurodevelopment disorder. And if any one of the component of these decorators and erasers are disrupted, so we see different neurodevelopment disorders in the forms of different types of syndromes. For example, if PHC1 is mutated in this PRC1 complex, it leads to primary microcephaly. If ring1 is mutated, ring1 leads to also very severe phenotypes of neurodevelopment disorder and schizophrenia. And so these are the PRC1 component, and TRIM37 is a ligase that is also involved in putting the ubiquitin mark And if TRIM37 is disrupted, it leads to multibrain nanism. And similarly, these autism spectrum disorder for OTS2. And similarly, towards my right, it's ASXL1, 2, and 3, which is if ASXL1 is disrupted, it leads to Boring-Opitz syndrome. ASXL2 causes Shashi-Pena syndrome, and ASXL3 causes Bainbridge-Roper syndrome. And USP16 causes Down syndrome. So yes, this sanity check we've made, like whatever experimentations we have done, it actually makes sense and it is contributing the knowledge to the functional validations and the genetic evidence for the ASXL3 gene. So what are the implications for genomic medicine? So for the genetic professional, the detailed understanding of supporting genetic evidence and functional validation enhances pathogenic variant classification. Whereas for the research focus, detailed understanding of supporting genetic evidence and functional validation informs model system choices. So now we know like ASXL3 is disrupting H2 monoubiquitination. I'll go in the lab, I'll make the brain organoids, and I will look for what are the cortical lamination defects, or if ASXL3 is disrupted, how it is affecting the neurogenesis, because microcephaly is also the one of the clinical feature of ASXL3 Bainbridge-Roper syndrome. So with this, I end today's lecture.